Okay, hi everybody. My name is Eric. Of course, you guys all know me. And for a while, I've been really wanting to make a video on COVID and the COVID vaccine and kind of the biology of it, just because not everybody has a degree in molecular biology slash microbiology, and I love talking about this stuff. And also, I feel like it's very important for everybody to understand the two of them, and then also, with the Delta variant kind of surging right now, I feel like hopefully this will help people make an informed decision about the vaccine. I have been meaning to make this video for a very long time, and I'm finally doing it just because really the Delta variant, it's extremely contagious. And if you haven't caught COVID yet, you probably will if you're not vaccinated. And so I'd really encourage people to kind of take this information and weigh the risks versus the benefits, especially given the new variants that are arising. So throughout this video, I'll just kind of talk a little bit about kind of cell biology and molecular biology and what mRNA is and how it works in our cells naturally. And then I'll talk a little bit about how viruses work and how they infect your cells. And then I'll also discuss the mRNA vaccines, which I recommend over the old vaccines, but of course, I guess I'll go over both of them too. So yeah, hopefully this is very helpful. And of course, I don't think that I could really speak to clinical trial data or safety data because that's not really my area of expertise. But I think that, just understanding how these vaccines work, and it's not just, this wonky serum that's being injected into you, and there's actually a reason that, everything that's in there is in there. Kind of really understanding the mechanism of the vaccine, I feel like is helpful for me at least. It really puts my mind at ease. I really don't trust pharmaceutical companies personally, and so I get why so many people are hesitant, especially if they don't understand the science of the vaccines. So hopefully you guys enjoy this. Hopefully this is helpful, and feel free to reach out to me with anything, any questions or concerns or criticisms of me, and I'll get back to you. So yeah, I'll probably just cut out or fast forward through the drawing part, but hope you guys enjoy it. Okay, so I drew a human cell, which there's a lot more to it, but I just kind of left out all the extraneous stuff, and it kind of looks like an egg, but just a quick label. So with each of your cells, except blood cells, I suppose, you've got a cell membrane. So this is kind of the coating that keeps the cell together. You have the cytoplasm, which is just kind of the goop inside of the cell. It's just the liquid that kind of or not floats, but houses everything inside of your cells, because your cells are not 2D, they're 3D, and so there needs to be something inside of them, and that is called the cytoplasm. Okay, and then finally, the last component that I'm going to talk about is the nucleus. So the nucleus is kind of also separated from the rest of the cell with another membrane. And the nucleus is where your DNA, your genome is held. So, every cell except blood cells, they don't have nuclei, have a little nucleus, and that's where they store their DNA, and then they send out their mRNA from the nucleus. And I'll talk about all that, but yeah, so nucleus is where the DNA is kept. All right, so I have to collect my thoughts. So essentially, in just day-to-day life, when your cells are functioning, when you're not dead, your DNA, your genome, which is housed inside of your nucleus, is being expressed, is what we call it. It's being used by your cell to carry out every function that cells do. So, say, I don't know, it's hard to think of an example that like everybody would know. Say that the cell gets too hot and the cell needs to cool down, or maybe not cool down, but it needs to be able to tolerate the heat that it's going through. Say that you're taking a very hot bath, which I like to do very often. My water bill is a little bit higher than it should be. But say that you're taking a hot bath and your cells are like, oh my god, it's hot. We're not used to this heat. We need to produce something to keep everything functioning. They have genes actually that are made to survive the heat. So they'll take that gene inside of the nucleus, kind of draw out our DNA. Take that gene inside of the nucleus, say that the gene is right here. It's encoded in this little chunk of DNA. They will take this DNA and they're going to copy it into messenger RNA. I'm sure that you guys have heard of that. So messenger RNA is what the vaccines are made of. Okay, sorry, I went on a little bit of a tangent and I cut myself off. Anyways, so we've got our kind of heat gene, our gene that's helping our cells survive when we're getting a little too hot. And so the cell, in order to express or use this gene, is going to convert it into messenger RNA. So let's say that the gene is this little chunk of DNA right here. So the cell is going to use what's called a RNA transcriptase and make basically a copy of the DNA, but it's made of RNA instead of DNA. So the difference is there's like a little chemical difference between the two. One of them has like an oxygen somewhere and the other one doesn't, and that's where the D versus the R comes in. So basically it'll copy it into the same sequence, the same DNA sequence, but it's in RNA rather than DNA. And RNA is just a temporary copy of the DNA. And I'll explain why we need a temporary copy of it in just a second. So, trying to think, like, I want you guys to be able to see it. It won't be cute, but I guess we can we'll do green. We'll do green for the RNA. Okay, so here's our RNA. So it's the same sequence, just single piece of RNA that was we call it transcribed from the DNA into kind of this new language of RNA. So that's why we call it transcribing, or transcription, because it's like somebody transcribing a book into another language. So the DNA is transcribed into RNA. And now we've got a piece of RNA of the gene of interest, the gene that's being expressed when I'm taking my hot bath. So this mRNA, this little piece of RNA, is going to be exported from the nucleus into the cytoplasm. So let's go ahead and export it into the cytoplasm. So now our RNA is hanging out in the cytoplasm. So the RNA, as I said, is really just a genetic sequence. The RNA doesn't actually do anything. It just codes for something that needs to be done. So how exactly does that get done? What happens is it's actually converted into protein based on the sequence of the RNA. So these little itty-bitty kind of organelles, we call them, or components of the cells, they actually latch on to the to the RNA, excuse me. And these little components are called ribosomes. And these ribosomes read the RNA, the messenger RNA, and convert it into proteins based on the sequence of the RNA. the sequence of these proteins then gives them different functions. So the proteins, or you could think of them as enzymes. You guys have probably heard the word enzymes. So enzymes are proteins that have catalytic functions. So they can make a chemical reaction happen, or they can make a chemical reaction happen. So these proteins aren't always kind of enzymatic. They also can have structural functions, like collagen is a protein, and it gives your skin the voluptuous, the hold that it has. Gelatin and gummies is what gives it kind of the structure, and when you bite into a gummy and it's got that great kind of squishiness, that's from a protein that was produced by a cell inside of a cow or a pig or whatever, wherever the gummy brand is sourcing their gelatin. So anyways, back to this. So we've got our kind of heat gene that has been exported or transcribed into mRNA, exported to the nucleus, or to the cytoplasm. And then once this mRNA is in the cytoplasm, ribosomes are going to convert it into protein. So I guess we can use brown for the protein. So this little ribosome is going to read the mRNA and convert it into a protein. And they really are just kind of like these weird little squiggly things if you look at them at like an atomic level. So I kind of like drawing proteins just like a little squiggle. So this little protein right here is going to actually carry out the function of protecting my cells from the heat. He's going to go and kind of latch on to different components of my cells that are sensitive to the heat and protect them. He's going to literally grab onto them and just keep them safe from the heat and from becoming too unstable from the heat. So he's going to just kind of grab onto them and stabilize everything. We call it a heat shock protein. So that's kind of the general gist of what's going on in our cells. We call it the central dogma of molecular biology. So you go from DNA, the DNA is transcribed into temporary pieces of mRNA, and then the mRNA is exported to the cytoplasm, where it is then converted by a ribosome into protein. And that protein is what actually does the function in the cell, be that keeping my cells safe from the heat or from the cold, or producing pigments in my skin or my eyes or my hair, or I guess the proteins actually may actually be the pigment in that case, producing the enzymes that actually come out of your sweat and actually help kind of kill bacteria and whatnot, different antimicrobial proteins. There's I don't want to get too technical, but proteins do literally everything when it comes to life on Earth. They do everything. So, I'm talking to you right now. Sorry, I'm just gonna. I'm talking to you right now because there are proteins in my lungs that are contracting, and in my throat that are contracting, little muscles, muscle cells in which proteins have been converted from DNA into RNA into protein, and they're actually carrying out a function of contracting my cells and helping me speak. So that's kind of the general gist of life. And so now I'll talk a little bit about viruses and how viruses work. So, and there's a ton of different ways that viruses work. They're basically just parasites on this process. So a virus, I would argue, is not a living thing, and it's actually just kind of hijacking your cells in this machinery that I've just shown you, this process, in order to replicate itself and infect new cells. So viruses are pesky little parasites on life as we know it, basically. And I won't get too into different life cycles of viruses, I'll just talk about COVID. So let's say that we have our COVID virus. Let's see, what color should I use? We will do purple. Yep, purple. Okay, so the COVID virus kind of has a membrane, just like your cells do. So I'm going to try and use the same colors for different components. So the COVID virus has a little cell membrane that actually came from the cell that it was created in. So it's literally a cell membrane around this around the virus, protecting it. The COVID virus has what are called spike proteins, which were, again, produced by the cell in which the virus kind of hatched and was created. So just add a couple lines, and that's our spike protein. Okay, and then inside of the cell, they have their viral genome. So the virus's actual kind of sequence, because when the virus kind of gets in here, it's going to hijack the machinery, and it needs a sequence in order to tell the machinery what to do. So with viruses, it's telling the cell to make more viruses, basically. So in order to do that, it needs to have a genome that codes for more viruses, and that genome can be DNA or it can be RNA. In COVID's case, it is made of RNA. So put a little, oh, just kidding, wrong color. A little RNA squiggly. So we have our COVID virus. So we have a cell membrane around the virus, we have little spike proteins, and then inside of the virus we have the viral genome, which codes for more viruses. So what happens is these spike proteins are going to bind to your cell, to the human cell. And typically I think that this is in throat cells, or like nose cells, but the virus can spread to other cells in your body, as we've found. So it can spread to sensory cells in your nose, so it can actually damage and kind of take over cells in your nose and damage those, and that's why people lose their sense of smell. It can, I don't know if we've shown that it can infect brain cells, but it does cause brain damage. But anyway, so that's beside the point. So you've got your human cell and you've got your virus. These spike proteins are actually going to bind on proteins that are present on the cell membrane. And in the COVID example, these proteins are, I think, called the ACE2 protein, or I don't really know, but there's proteins on the cell membrane that are going to bind the spike protein. So just draw a little line between these two. I know this is going to be a really long video. I'm sorry, people. Oh my god. Okay, so kind of drew the next step. So you had your virus with its spike proteins, you've got your human cell with its ACE receptors on it, and the spike proteins are going to bind to the ACE receptor here, and then that's going to actually trigger the cell entry of the virus. So it's actually going to trigger the cell to let the virus into itself. And the reason it does that is because it doesn't think that it's actually a virus. It thinks that it's some other, could be food or something else. basically it's just eating the virus. So I'll draw that as well. Okay, so now you can see here, so virus and cell, the virus binds to the cell through these kind of membrane, these proteins that are attached to the membrane of your cells. And then the cell will almost kind of start to swallow the virus. And then I'll draw it here in a second, but the virus will then kind of enter the cell and be kind of free floating inside of the cytoplasm of the cell. Okay, so now the virus has entered your cell, and you kind of have this weird situation where the virus is totally entombed by the cell membrane. So it literally just kind of like buds into the cell and creates this little pocket of membrane. And this is a normal process actually. So cells do this to obtain nutrients and to get things into the cells that wouldn't normally be able to get across this little membrane very easily. And so this is a natural process that your cells are doing all the time. There just so happened to be a virus this time that got into the cell, dang it, and now your cell is infected. So this kind of encased virus is actually going to break free. And these two kind of membranes are going to actually like fuse together and kind of let the virus out. And by virus, I mean the viral genome. So I'll draw that really quick. Okay, so now we've kind of got our last stage of the kind of viral entry, the infection pathway. So when you breathe in a virus and it hits your throat cell, this whole process is going to happen. So you start with the virus binds to your cells, the virus is then kind of engulfed by the cell, and then finally the viral genome is released into the cytoplasm, the viral RNA. And this RNA, I'm sure that you can guess what happens with it when you've got some RNA floating around in your cytoplasm and your cell is used to kind of turning that into protein. The cell is going to turn it into protein. So the cell is going to start making copies of the virus itself. Okay, sorry, I had to check my notes to make sure that I was doing the virus life cycle properly because there's so many of them. I don't want to get it mixed up. So you've got your viral RNA, it's been released into the cytoplasm, and now the cell is starting to take that RNA and actually turn it into more viruses. So that this RNA right here actually codes for more virus. So your cell is just going to start pumping out viruses based on the code of this RNA that was in the virus itself. So just kind of draw that out really quick. So you've got some ribosomes that latch onto the viral RNA and start converting it into different proteins involved in kind of the structure of the virus. I'm gonna actually erase the central dogma. I think you guys probably got that already, and I need a little more space, so I'm gonna erase that. All right. So your cell's going to start kind of turning this into different proteins, and for the sake of this, we'll just talk about the spike protein. So your cell is going to start turning that into protein, turning that into more viruses, and it's going to convert that into more spike proteins. And those spike proteins are actually going to be kind of sent to the cell membrane, and they're going to chill in the membrane for a hot second while the virus replicates itself inside of your cell. Okay. In addition to making more spike proteins inside of your cell, the RNA piece of the virus is actually going to replicate itself using enzymes that were kind of packaged in with the virus itself. And I didn't include that just because I feel like it's already complicated enough. But basically the cell is going to be instructed by this viral, the contents of the virus to make everything that's necessary to create more viruses. So that includes more viral genomes and more spike proteins, and then more other stuff that I don't include here. So for the sake of this, let's just say, okay, so the cell produced a bunch of spike proteins based on the viral RNA, and it also copied the viral genome into a bunch of different pieces of viral genome. So now there's a ton of viral genome in the cytoplasm, and there's also spike proteins that are chilling on your throat cell membrane. These are actually going to then package and do kind of the reverse of what we just saw here. So they're actually going to bud outside of the cell and then turn into new viruses. So these kind of little pieces of viral genome are going to be packaged into individual viruses and sent off into the rest of your body to infect new cells. Or you're going to cough them out and you're going to infect somebody else. And then this process starts all over. So I'll draw that out really quick. I'm going to erase kind of the labels here for everybody, just so that I have a bit more room. Actually, no, we're just going to move this up here. So I'm just going to repeat what's drawn right here, up here, and we'll use some of the space up here. So got all of our copies of the viral genome as little pieces of RNA. Got all of our spike proteins that have been created from the RNA and sent to the membrane. This RNA is going to be packaged and kind of sent outside of the cell as a new virus by your own cell, actually. So as I've been saying, viruses are basically just parasites on life. They just take over cells with genetic information that harms the cell and kind of devotes all of its resources towards creating more viruses, and then those go out and infect new cells and do the exact same thing to those, and then so on and so on. So draw that really quick. So you've got your viral RNA inside of the new budding virus, and you've also got your spike proteins. And then that's going to be converted, or once it's done kind of budding out of the cell, you're going to have a fresh virus. And that is going to go off and find new hosts. Okay, so that's the viral life cycle, and that's how COVID infects your cells and then replicates and then spreads throughout your body. And then if any of these viruses just so happen to get into your saliva, which many of them do, and you cough, then that might infect the next person, person sitting next to you in the coffee shop or at the grocery store. I think now is actually a good time to talk about the Delta variant. So these variants that are popping up all have mutations in their spike proteins. So as I mentioned earlier, the spike proteins actually bind to these ACE receptors on your cells, these proteins that are just kind of sticking out of your cells. And they have some function, but I don't really know what they do. But so the mutations in these new variants are actually in the spike protein itself. And the mutations cause the spike protein to bind even stronger to those ACE proteins that are on your cell membrane. And so many times, cells will, if you happen to walk by somebody in the grocery store with the original COVID variant, and that person was infected, you might get, a few viruses in your throat, but, some of them might even get to the point where they bind onto your ACE receptors for just a hot second, but they don't bind strong enough, and they kind of dissociate from your cell membrane. They, bounce off and go into your stomach, and your stomach acid just destroys them, and, you're safe, you're fine. But with the Delta variant, these spike proteins are started to mutate to bind even better. So they're evolving to bind even stronger to our ACE receptors. And so say that you're passing somebody at the grocery store and you breathe in a few viruses from them, there's a much stronger likelihood that, when that virus comes into contact with an ACE receptor, it's actually going to bind and it's going to stay there until it's inside of your cell and has infected your cell. So that's why that's why the Delta variant is very scary. And I think also the other thing is that it has also mutated, the viral genome has mutated in such a way that the cell is instructed to produce many, many more, like magnitudes more virus. And so you're actually shedding a lot more virus when you're infected with the Delta variant. So instead of creating, one cell creates 100 viruses, you're now creating 10,000 viruses from one cell or something like that. Those two put together are why the Delta variant is very contagious. Also, I want to say, if you're still listening to this, congrats. This is long. This is really long. But it's hard to kind of boil this down in such a way where, I'm telling and I'm explaining the science of it, but I'm not dumbing it down to the point where you just have to trust me. I would rather just show you the whole thing, and maybe get a little bit more trust in what I'm saying, when I say that, I could just be here and say, mRNA vaccines are, just what's happening in our cells all the time, and it's not a big deal. But if I actually go through and show you kind of how they work, it is a little bit less, or a little bit more transparent, I guess is the right word for it. So this is our viral life cycle right here. And so the mRNA vaccines, the way that they work, it's actually going to be really quick for me to explain this now that I've done all the heavy lifting of explaining life as we know it in viruses. So the mRNA vaccine, for this I'm actually going to erase the labels that I have just because I feel like it's nice to have all of this here. So again, nucleus, cytoplasm is just the insides, and then the cell membrane is the membrane that keeps it all together. Okay. Okay. So the vaccines are made of mRNA, and the way that works is kind of the mRNA gets into your cells, and then the mRNA codes for the viral spike protein, and so your cells will kind of produce the spike protein inside of themselves. They'll send it to the membrane, and then your immune cells will recognize the spike protein and be like, what the hell is this? I've never seen this before. There's probably a virus in this cell. Let's destroy the cell and also, take this little thing that's on the membrane, this little spike protein, and let's kind of replicate it and then produce antibodies against it. So, so basically, how do I want to start this? Yeah, okay, so in the vaccine, what you've got is a little membrane made of little fats, just like our cell membrane is made of little fats. So you've got a little membrane, and then inside of that membrane, you have a little piece of RNA, messenger RNA, that codes for the spike protein in the virus. Okay. Oh my God. Okay, so, oh my God, my markers keep falling off the table. So, when you're injected with the virus, with an mRNA vaccine virus, or no, there's no virus in the vaccine. When you're injected with the mRNA vaccine from Pfizer or Moderna, what you've just been injected with is a bunch of little membranes full of RNA that has the spike protein inside of it, or the spike protein RNA, the code for the spike protein as messenger RNA. I'm tired. I worked 9 hours today, and I haven't had much to eat, and yeah, it's been a long day, but I wanted to do this and just get it done, especially seeing as things are getting bad again. So, been injected with the RNA for the spike protein. This little membrane is actually going to fuse with the cell membrane of the cell, of, many cells in your body. And where's my marker? I lost my marker. Oh, there it is. Okay. Yeah, and then that RNA is going to enter the cell, and it is going to be converted by the ribosomes in your cell into spike protein, even though your cell has not been infected with the virus. So we've got our little ribosomes. They're going to convert the spike protein code from the vaccine into actual spike proteins. Here, I'm going to draw a little line so you know where the virus stops and the vaccine begins. Okay. Or we'll do it up here too, so that we can have some immune cells coming in here. Okay. Then you're gonna have an immune cell that just happens to walk by, and he's like, oh my god, what is that? That looks like a viral spike protein. Give him a little smiley face. Or actually, no, he's not happy because he just found a spike protein. He's unhappy. Oh my god. So he's alarmed because he just found a spike protein. And he's gonna kind of tell this cell that, okay, you're infected, you need to go. And he's either actually gonna eat the cell, or he is going to kind of trigger the cell to commit suicide. I believe is typically how it works. And then he's going to kind of take these spike proteins and he's gonna make antibodies against it. And he's gonna kind of save the information on how he made those antibodies, and he's going to give that to other cells, and he's going to kind of turn the body into an antibody producing factory. Now that it's been exposed to the spike protein and it's producing antibodies against the spike protein, what will happen is instead of, so say you walk by somebody in the grocery store, and again, you inhaled a few viruses from them because they were infected, you are now teaming with antibodies, which we will make purple, or kind of this weird mauve color. So you are now teaming with little antibodies that all bind to the spike protein in the virus, or on the virus rather. And so, instead of binding to the kind of ACE receptors on your cells, these viruses are going to be grabbed onto by the antibodies, and they're going to be kept from binding to your cells, and then they're just going to, wash down your throat or wherever, they'll be caught in your mucus, and you'll, sneeze it out or something. But they won't be able to bind to your cell and actually get inside of your cell, because they're literally going to be like handcuffed by all of these antibodies. Oops. So they're just going to be surrounded, their spike proteins are just going to be coated in all of these antibodies. They're not going to be able to bind your cells, and they're just going to wash away, basically. They have been neutralized. Is how they put it. So, and then with the kind of old versions of the vaccine, typically what will be done is they will produce the spike proteins in, yeast or bacteria. So just like, our cells are able to make these spike proteins when they're given the instructions for it, scientists in a lab will go in and they will instruct, actually genetically engineer sometimes the yeast or the bacteria to produce spike proteins. They'll grow the yeast and these bacteria in these huge vats, and then they will kill the bacteria or the yeast or whatever, and purify the spike protein, and then inject the spike protein into you. And that way you've got just spike protein like in your blood, and your body's gonna be like, the hell is that? We need to make antibodies against that. Another option is they will, I think that this is how the Johnson & Johnson and the AstraZeneca vaccines are made. They will use, so it's similar to this, where it's kind of just like a membrane that fuses with the cell and kind of delivers the code for the spike protein. But with the AstraZeneca or Johnson and Johnson. I think that this is the correct technology for it, but I'll edit this out or I'll change it at the end if I'm incorrect. But basically, they use a virus, just like COVID, but the virus is an adenovirus. It's a different kind of virus, and it's been inactivated, and the only thing inside of the virus, so instead of coding for the entire virus itself, the virus only codes for the spike protein. So viruses are just really great at getting, genetic information into our cells. And so with the AstraZeneca and the Johnson and Johnson vaccine, they use kind of a different kind of virus that will bind to your cells in a similar way, where it'll bind to little receptors on your cell membrane, and it'll actually like shoot the DNA into your cells, the RNA, not DNA. Nobody's being injected with DNA. So this like fake virus, this other virus will like shoot the spike protein RNA into your cell, and then your cell will produce the spike protein, and it'll be the same thing here, where your immune cells will be like, the hell is that? And come and make antibodies against the spike protein. So so kind of the AstraZeneca and Johnson technologies, they're kind of the last generation of vaccines, and these mRNA vaccines are kind of the new generation. And they've proven to be very effective and very safe, just based on what the public health directors say, and very effective just because of all the data, all the academics and all the public health directors, and everybody that's apolitical. I promise you that people like me who are just academics and scientists, we just want the best for everybody. We're not trying to genetically engineer people or make money. I promise you that unless you work for like a pharmaceutical company, and a lot of times even if you work for a pharmaceutical company as a scientist, you don't make a lot of money. So we really don't have an incentive to lie about this stuff. I hope that this was helpful. Please reach out to me if you had any clarifying questions or you wanted to know more. Thank you. So just some quick edits, because I don't want to retake that like hour-long video. I said RNA transcriptase when I was talking about converting DNA into RNA. That's actually done by an enzyme called RNA polymerase. I'm like kicking myself because that's like a stupid mistake. And then for the Johnson and Johnson and the AstraZeneca vaccines, they actually they do use an adenovirus. I was correct about that. It doesn't shoot the RNA into you. It's very similar to the way that COVID enters your body, or your cell. So it'll kind of bind and then kind of be internalized by the cell, and then it'll break out of that little pocket that forms. Again, like there's no DNA or anything like that involved. And something that I kind of forgot to touch on is that RNA cannot be converted into DNA, so don't worry about like being genetically engineered or something like that. It's not going to happen. It's impossible for a human cell to be able to convert RNA into DNA. Like there just isn't the machinery to do it. I hope that this was informative and helpful. And again, please reach out to me if you guys have any questions, or you want to yell at me for being, for talking about COVID or something. But thank you guys, and hope you liked it.